Hello, this is Dr. Rhonda Fellheim. I guess you can tell from looking at this slide what we're going to be talking about, and you can probably also tell where our formed elements get their names from. Yep, white blood cells, red blood cells, and platelets. I love this picture. This is just a guess what this is kind of picture. Pretty amazing. These are red blood cells or erythrocytes on the tip of a needle, which I think is pretty cool. Okay. The function then of blood, as we just talk about blood, there's several functions that we talk about, and transportation falls right at the top of the list because we know it transports very important gases that keep us alive. It also transports nutrients, hormones, and ions, any minerals, other substances that we need in order for our cells to stay alive. Regulatory. Our blood actually participates in the regulation of pH of the fluids in our system, and blood volume, and that has some far-reaching effects. And then protection, meaning the leukocytes, or the white blood cells, participate a great deal in protecting us from things that are foreign substances that would harm our homeostasis or disrupt our homeostasis. And so protection is a real very important role of blood. When we look at the composition of blood, if we took a tube of blood that we just drew and we spin it in a centrifuge, the heavier formed elements would accumulate on the bottom, and these are the red blood cells or the erythrocytes. You then get kind of a buffy layer or buffy coat, which is just the leukocytes or the white blood cells, and then you get the plasma, which is the fluid and its solutes. When we talk about what percent of the whole blood is made up of the red blood cells, we call that hematocrit. And I'm sure that most of you have heard people ask, "Oh, what was their hematocrit?" Or even on a TV show. This is just another great picture of blood. There are some amazing pictures of blood out there, and we really, I don't even walk around thinking that my blood looks like this. That's just that cool. Okay, so let's break whole blood down. It's only about 8% of our body weight. Let's break that down into its components. And I know this picture looks a little daunting, but we'll break it down into its parts. The plasma, again, is the fluid and its solutes, and that's, of course, primarily water. We have a significant amount of the solutes are protein, and the proteins are made in the liver. The most abundant protein is the albumins, and this category, we know that they are co-transporters. They affect the osmolarity of the blood, and moving on to globulins. The immunoglobulins are the only ones that really aren't going to be made in the liver. We'll learn more about those when we talk about immunity, but the other globulin types of proteins are made in the liver, and they make up a number of structures or components of structures in our body. Fibrinogen is a clotting protein that we will learn is in our blood in an inactive state until activated, and very much participates in clots. Some of the other solutes that we have in our plasma are, of course, electrolytes, nutrients, gases, the things that we talked about earlier that are transported in our blood. Okay, and again, very important, not only are we taking things that will keep our cells happy, not only are we transporting that, but we're also transporting waste away from cells. When we talk about formed elements of our blood, we're talking about the cells. We're talking about the red blood cells, or erythrocytes, the white blood cells, or leukocytes, and then the small little platelets. And we can see that when we look at volume-wise, you know, over 5 million red blood cells per liter. And when we look at the other numbers, you can see, well, about, somewhere up to 400,000 platelets, or just less than 10,000 white blood cells. So platelets, as you know, another word for platelets is thrombocytes, and so we will talk a bit about those. And when you hear the word platelet, I'm sure that you can think of what it is they do. Most people would respond with, they participate in clotting. Okay, good. When we talk about white blood cells, leukocytes, we've got 5 different types of, or 5 individual types of leukocytes, and they're very much going to be involved in different aspects of keeping us healthy. So we will, you can see from this table how they appear number-wise in our blood. You can see that neutrophils are by far the most abundant. That we then see numerous lymphocytes, and then the numbers drop significantly as we move from monocytes, eosinophils and basophils. One of the reasons that we don't see many leukocytes in our blood is because they are very, very abundant in lymphatic tissue, and so that's something that we will explore further when we talk about lymphatic tissue. But we do see some of these in the blood. However, again, they love to live out in the tissue, in lymphatic tissue, and plus that's where they work the hardest and do their job very effectively. So if I were to ask you, just from our little discussion here, what's the important role of albumin in the plasma, take a look. I would say D, colloid osmotic pressure, if these were my choices. Okay. This again these are red blood cells, or erythrocytes, that we see under an electron microscope in a capillary. And you'll see in just a second why we would be seeing this particular shape. All right. Well, if we look at, there's that shape. Doesn't that look familiar? So we were looking at red blood cells on end. Okay. Now, a red blood cell is an erythrocyte. Okay. Erythrocytes are red blood cells. Same thing. And so I have a tendency to interchange the two. But we look at this cell and we say, what words would we use to describe this cell? Well, typically it's described as what we call a biconcave disc. And biconcave disc then gives us a whole lot of surface area for gas diffusion, for gas exchange, and we're going to know that that's very, very important, and that's a key job of the red blood cell. So it does increase our surface area significantly. Now, one of the other things you'll notice is that there isn't a nucleus in the red blood cell. And so I want you to give some thought to, what does that mean in terms of what the red blood cell is not capable of doing? And you're going to have to search through the cobwebs of 2085 material and think about what it is that nucleus orchestrates. What is that? How important is that information in the nucleus, and what are cells able to do because of that information? Well, let's look at what is in a red blood cell. What is in a red blood cell is it's jam-packed full of what we call hemoglobin. Hemoglobin. Now, the word globin refers to protein structures, and the word hemo, of course, is blood. And so this is one molecule of hemoglobin. And we can see that one molecule of hemoglobin then is made up of 4 globin proteins. All right? And each globin protein then has a heme group in the middle of it. This pinkish sort of thing is a heme group. Well, let's look closer at the heme group, and then we'll come back to this. Let's look closer at the heme group here. This is the chemical structure of the heme group, and no, I don't expect you to memorize that. However, I do want you to recognize that there is an iron molecule in the middle of the heme group, or close to the middle, and that it's at this iron molecule that the oxygen molecule will bind. So oxygen binds to the iron within the heme. All right, oxygen molecule binds to the iron within the heme. So if we go back and we say, oh, so for each molecule of hemoglobin, we have a heme group, and each heme group is going to be capable of binding one molecule of oxygen. How many molecules of oxygen can a hemoglobin molecule bind? All right, the answer to that is 4. Yes, you got it correct. Good. If you did not get 4, you should go back and review those slides that we just covered. Okay, that will help to clarify that. When we talk about hemopoiesis or hematopoiesis, those two words are going to mean the same. And essentially what they mean is the production of blood cells, of formed elements, the production of formed elements. And so then let's look just more specifically at what we call erythropoiesis. I always feel like I'm speaking a foreign language when I talk about this particular slide, because every word is a variation on the others. Okay, so let's talk about erythropoiesis, the production of erythrocytes, or red blood cells. Okay, so we start with what we call the hemopoietic stem cell. And again, when we say stem cell, they're typically capable of differentiating into different types of cells. And so we will see that this same stem cell is capable of differentiating into leukocytes, or white blood cells. But here, what we're talking about are the process that will result in us having a mature erythrocyte in the blood. So erythropoiesis then is this process, and one of the things we want to look at is, okay, well, what sets this process into motion? What sets this process into motion? Well, the word would be hypoxemia. Hypoxemia. I know I have hypoxia here, but it's hypoxemia. And that is low blood oxygen levels. Okay, low blood oxygen levels. That's what hypoxemia means, emia. And so when low blood levels of oxygen are detected at the kidney, okay, so this occurs at the kidney picks up on the fact that the oxygen levels in the blood are low, and it will release an enzyme or hormone. This is called both in different references. So it will release a hormone or enzyme referred to as erythropoietin, or erythropoietin, EPO. Now, the erythropoietin is actually going to bind on the erythrocyte colony forming unit, on this particular cell here. Okay, there are binding sites for that EPO on this cell, and it will lead to then the cell increasing in number, making a whole lot of themselves, increasing the production for these cells that are down the line. Okay, so the erythropoietin then has its effect here. So as we come down the line then of this process, we reach this cell called the reticulocyte. Now, it's actually the reticulocyte that's going to be released into the blood. This is our immature red blood cell. Okay, and what we see here in this, because of the blotchy look, those are actually ribosomes, and but you don't see a nucleus, because at this stage we lose the nucleus. Okay, so the nucleus gets lost here. We then have a reticulocyte which just has some ribosomes left in it. The hemoglobin is already there. That happens over here. So At this stage then, hemoglobin production and losing the nucleus. What's left? Ribosomes in the reticulocyte. All right. This is our immature red blood cell. It is released into the blood. And we expect to see a certain number of reticulocytes in our blood. So they will mature into erythrocytes over the course of just 2 to 3 days. Essentially, they will lose the ribosomes, and we will then have a mature. Now, we know the life of an erythrocyte is a rough one. They get thrown out of the heart with a great deal of pressure behind them. They travel all over the body. They actually squeeze through capillaries, because most capillaries are actually smaller than the smallest of capillaries are actually smaller than red blood cells. So they will squeeze through. They'll fold over. They'll and so they get beat up pretty bad. The average life of a red blood cell is about 120 days. And so, the bottom line, we know that without a nucleus, I'm giving you the answer to a previous question, without a nucleus, they're not going to be able to repair themselves. The template is not there. The DNA is not there, the genetic information, in order for them to make proteins or in order for them to mend their membrane. So when they get beat up or they get damaged, they have to be taken from the blood. They are removed from circulation. This primarily occurs in the spleen and liver. And the spleen does a big part of that, just pulling them out of the blood. And then they're consumed by macrophages within those organs. So again, about 120 days. And that's why when we talk about erythropoiesis, it's a pretty amazing process, because we dump about 2 million new reticulocytes into our blood every second. So things are happening in a very big way. So the thing about blood, the thing about red blood cells, is that we can't just break it down into its parts and just let it be, because we do recycle. We are big on recycling, and some of the parts, though, can be quite toxic to us, so we have to actually get rid of them. So let's take a look at what do we do with a red blood cell when we break it down. Okay, the globin part, the protein part. Well, we know our body can handle proteins readily, and so we just break those down into amino acids, and then we use the amino acids in our other cells, making proteins. That's no problem. However, that heme group has some controlled substances in it that we need to get rid of. Okay, first of all, we do treat iron as a controlled substance because bacteria thrive on iron. They really like iron a lot, and we don't want bacteria doing that in our bodies. So several things we can do with that iron. We can either attach it to a specialized transporter and have it escorted to where it's going, which would be either the liver for storage or to the bone to be put right back in blood. Okay, and we know that blood is made in the bone marrow. That's where that's going to occur. So that's where hematopoiesis occurs, in the bone marrow, red bone marrow. And so this iron may go back to the red bone marrow and show up in new blood, or sometimes we will lose iron through injury or menstruation. Now, the rest of that heme group is converted to a substance called biliverdin, when this within the liver. It is then converted to a substance known as bilirubin, and we then get rid of the bilirubin oftentimes through our feces in a substance that the liver produces called bile. We'll learn a whole lot more about bile when we talk about the we definitely try to get rid of this through our feces. Now, there is a conversion that sometimes takes place of the bilirubin into another substance that can be released in our urine. But again, it's just another pathway for getting rid of it. Okay. But that's what happens once we degrade hemoglobin molecules. Now, we can look at this process as well, the leukopoiesis, and you probably can see by now that this is, or you would understand by now that this is the production of white blood cells, leukopoiesis, producing leukocytes. And again, this is happening in the red bone marrow as well. But look what we start with. We start with that same hemopoietic stem cell that we started with the red blood cells. Okay, so they're capable of differentiating then into the different individual leukocytes. When we get to this point, you can see that the lymphocyte is actually going to, the three different lymphocytes actually come from their own precursor cells. However, we ultimately started at the hemopoietic stem cell. Okay. Now, in lab, we're going to spend a lot of time talking very specifically about the different leukocytes, how to differentiate between them, what do they look like, what's their function. So we'll do that primarily in lab. We will talk extensively about lymphocytes when we talk about immunity in the lymphatic system. So I'm not going to go into much more detail than I already have. So I'm going to end this discussion now.